Welcome back to MicroVerse. In the previous lecture, we discussed why the classical Koch postulates are not always applicable. We saw that some microorganisms cannot be grown in artificial culture. Some pathogens have complex relationship with their host, and disease causation may involve multiple microbial or host factors. This created a new challenge for microbiologists. Instead of asking only which microorganism caused the disease, modern microbiology began asking a more specific question: which microbial gene or molecular factor actually contributes to disease? This led to the concept of a molecular Koch postulate proposed by microbiologist Stanley Folka in 1988. From microorganism to gene, classical Koch postulate mainly connected to the microorganism to the disease. Molecular Koch postulate take this one step further. Virulence gene to virulence phenotype. The focus is therefore no longer simply on identifying the pathogen. The objective is to determine whether a specific gene or gene product contributes to the ability of microorganism to cause disease. The first principle which state the first principle is that the phenotype or virulence trait under investigation should be associated with the pathogenic strains of the organism. For example, suppose a bacterial strain possesses a particular factor that helps to adhere to host cells. If that factor is associated with pathogenic strain, it becomes a candidate virulence factor. So the first step is essential: identify the suspected virulence trait. The second principle is the next step of an experiment. Scientists specifically inactivate or delete the suspected gene. Then they compare the modified microorganism with the original strain. If removal of that gene produces a measurable reduction in a pathogenicity or virulence, this provides evidence that the gene contributes to the disease causing phenotype. So remember, gene remove means virulence decreases. This is the key experimental idea. The third principle, but there is an important final step. Scientists then reintroduce the normal or wild type gene into the mutant microorganism. If the original virulence phenotype is restored, the evidence becomes much stronger that the gene was actually responsible for the observed effect. Therefore, gene removed means virulence decreases, and then gene restored means virulence returns. This restoration is known as genetic complementation. The complete logic. So instead of memorizing complicated statement, understand the experimental sequence, identify, inactivate, observe loss, and restore. Or even more simply, gene remove, virulence decreases, restore, virulence increases. That is the central logic behind molecular Koch postulate. So why is this important? This approach allow researchers to move from simply identifying pathogens to understanding how pathogens cause disease at the molecular level. It helps scientists investigate important features such as adhesion, invasion, toxin production, and immune evasion. These are examples of microbial properties that can contribute to pathogenicity. Molecular Koch postulate therefore become an important conceptual framework for studying microbial virulence and host pathogen interaction. So, however, we should not interrupt molecular Koch postulate as an absolute rule. Virulence is often multifactorial. A microorganism may use several genes that work together, and different many genes may compensate for one another. Therefore, deletion of a single gene does not always completely eliminate virulence. Modern microbiology, therefore, uses molecular Koch postulate as a framework for establishing causation rather than as an inflexible checklist. So, here I give you a one memory trick. Remember, find, knock, loss, and restore. Means find the virulence associated gene, then knock out the gene, then lost the virulence phenotype, at last restore the gene and virulence return. That's the entire concept. The significance of this concept extend beyond the examination questions. Modern microbiology uses molecular and genetic approach to understand virulence mechanism, identify potential therapeutic targets, and investigate how specific microbial factors contribute to disease. This represents an important shift in the microbiology from identifying the pathogen to understanding the molecular mechanism of a pathogenicity. So, what did my molecular Koch postulate at the classical Koch work? They changed the question from which microorganism caused this disease to which microbial factors make this organism capable of causing disease. That shift from the organism level to the gene level was an important step towards modern molecular microbiology. So, we have now followed the history of microbiology from the discovery of microorganism to molecular investigation of their pathogenicity. But microbiology did not stop with understanding which organism causing disease. The next major challenge was to understand how this microorganism interact with the human host and produce disease. That brings us the next major concept in our journey, pathogenicity and virulence. So thank you for learning with the MicroVerse. Keep learning, keep growing, and let's master microbiology, one concept at a time.